Hello, my name is Itzayana Alfaro and today I will be talking about mitosis and meiosis. So let's begin. Mitosis. These are the different stages that it goes through. So we'll go ahead and start with the G1 phase, which is also known as the starting or the growth phase. The cell grows physically larger, it copies organelles, and makes the molecular building blocks it will need in later steps. In the S phase, that's where the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates a macrotubule organizing structure called the centrosome. The centrosomes help separate DNA during the M phase, and we'll go more in detail in just a second. In the G2 phase, the cell grows more, it makes protein and organelles. It also begins to reorganize its contents in preparation for mitosis. Once the G2 phase ends, that's when mitosis begins. During the M phase, the cell divides its copied DNA and cytoplasm to make two new cells. So let's begin with mitosis. In the prophase, which is the first step, that's a stage where the meiotic spindle is beginning to form and the nucleolus is disappearing. In the prometaphase, right here, is where the sister chromatids are fully formed, as you can see right here. The chromatids are attached to the spindle by the kinetochore. The nuclear envelope has disappeared by then. As you can see, there's no more nuclear envelope. In the metaphase stage, which is right here, that's where the sister chromatids are aligned in the center, right here in the metaphase plate. The microtubules are attached to the centromeres. As you can see right here, they're in contact with the centromeres. And that makes it be able to go to the next stage, which is the anaphase. The anaphase stage is where the chromatids move to the opposite ends of the cell. As you can see, they start pulling apart. And that makes it be able to go in the telophase and the cytokinesis stage. So the telophase stage is where the chromatids are at the opposite poles. As you can see right here, they're no longer pulling apart, but now they are actually apart. The nuclear envelope forms again. As you can see right here, it's starting to form all around. And the chromatids decondense, the aster breaks down, and cytokinesis begins. As you can see, the cytokinesis is starting to begin because it shows the cleavage. The cytokinesis is when finally you have two new cells formed. So now we will continue talking about meiosis. Meiosis is separated into meiosis 1 and meiosis 2. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, telophase and cytokinesis 1. As far as meiosis 2, it goes with prophase 2, metaphase 2, anaphase 2, telophase, and cytokinesis 2. So let's go more into detail with those. As you can see here, for the first phase of meiosis, it's referred as to the prophase 1. In this stage, the chromosomes condense. As you can see, this is how it starts. They start condensing. The nuclear envelope disappears. Right here, you can see it. And crossing over occurs in this phase. As you can see right here, it says crossing over. So they're crossing over information, and this comes into this result. As you can see, there's a blue right here, and then there's red over here where they exchanged already. Now metaphase 1. In metaphase 1, the pairs of the homologous chromosomes move to the equator of the cell. The equator will be the center of the cell. Now for anaphase 1, the homologous chromosomes move to the opposite poles of the cell. As you can see, it's starting to separate, and we see a little bit of cleavage right here. Now for the telophase and cytokinesis 1. In this, the chromosomes gather at the poles of the cells and the cytoplasm divides. As you can see, right here, they're starting to divide more and more. So that makes it to be able to continue to the next stage, which is the prophase 2. In the prophase 2, a new spindle forms around the chromosomes, as you can see right here. That makes it be able to continue to metaphase 2. In metaphase 2, the chromosomes line up at the equator of the cell, just like it did before, but now, as you can see, there's actually two cells, not just one. Now there are 2 cells. And then anaphase occurs. The anaphase 2 is where the centromeres divide and the chromatids move to the opposite poles of the cell completely, as you can see right here. Let's go back to the metaphase 2, and they're together. Now for anaphase 2, they're being pulled apart. And that makes it be able to finally reach into the final stage of meiosis, which will be the telophase 2 and cytokinesis. By then, a nuclear envelope has formed around each set of chromosomes and the cytoplasm divides. As you can see, the nuclear form right here. The cytoplasm has divided, and you can slowly see how it's not 1, 2, it's 3, and it's 4 daughter cells. Thank you for watching my presentation about mitosis and meiosis. I hope you enjoyed, and I also hope you learned. Thank you.